Okay, so good morning and a very warm welcome to this innovation workshop sponsored by Pharmidex on the topic of drug discovery solutions. So I'm delighted to be joined by a range of experts from Pharmidex this morning, and I'll shortly introduce them. Ahead of that, just to clarify that this innovation workshop is being recorded and will be available on the conference app post the event for 4 weeks. For delegates to view at their own convenience. And the chat box is enabled, so we welcome any questions or comments from those online. So joining us online this morning, we have Ash Alavijay, who is Head of Operations, Andy Harris, who is Head of Bioanalysis, Ian Knowles, who is Head of Respiratory Research, and Steve Mitchell, who is Head of Integrated Therapeutics. So a real expert lineup. So I'm really looking forward to the insights that the panel has to share. So I'm going to hand over now to the Pharmidex team to guide you through their presentation. Over to you guys. Thank you.
That's great, Alicia. Thank you very much for introducing us. So this is a Pharmidex presentation for the Helix program. So I'll be starting the presentation today and I'll be passing on to my colleagues as we go along. So a little bit of background about the company, so about us. So Pharmidex we're a small organization, we're a CRO founded in 2002. So we've got a collaborative R&D program, we work with lots of different companies. We've got 30 plus staff at the moment. I think we're currently at about 42 full-time members of staff. We work with up to about 300 clients, which are pharma, biotech, virtual academia, and medical charities. And these clients are worldwide. So not only do we work with the UK and the US, but a lot of Western Europe, Far East, and all around the world, really. And just to go into a very brief history about the company itself, so we founded in 2002. We had our first round of investment by a medical device company in 2004 called FlexiCare, who are very much still with us. In 2007, we also had some VC funding from a company called A2E based in Manchester. And we also had a strategic alliance with a company called Genzyme, which has now been acquired subsequently. 2010, we worked with another UK-based company in medicinal chemistry called Domainix, and we still very much have a strong partnership with that company. We moved on in 2013 to SBC, as you guys might know, which is the Stevenage Biocatalyst, which is based inside GSK. So we still have a presence there to this day. And in 2016, we found our more permanent home, which is in the EKC, which is based in Hatfield, which is an ASI site. So all our lab operations are currently based within this site, and we'll be here for the long-term future. 2017, we opened an office in Switzerland. Obviously, with everything going on in Europe, we wanted to tap into that market, and that's worked quite well for us. 2018, we nicely moved on to acquiring a company called Newer Labs, who were specialists in respiratory work, and that'll be covered by my colleague later on inside the presentation in more detail. So 2020, we also got our MHRA GLP accreditation. In 2022, we've got our 20-year anniversary, which just passed by. I'm proud to say that this year we're going to get our ADAC accreditation as well, which hasn't quite made a presentation, but that's something nice to follow on from later on. In terms of general accreditation as well, of course, as a UK-based company, we are Home Office compliant. Of course, we have multiple licenses that let us occupy and do certain procedural work in the UK. We have our MHRA accreditation as well, so we can do regulatory work, so that's GLP and GCP analysis as well, and GLP tox, of course. In terms of NIH, we have coverage from the US facilities as well, so we're able to do grant-funded US work, which is covered by our accreditation here as well. And of course, we have OLA as well, which is another US one, which covers our welfare standards up to a certain date, which is fantastic. On this topic as well, we're also going to get ADAC accreditation this year as well, so it'll give us full coverage within the western side of Europe and also the US market, so we're able to do all different types of work for different clients under those accreditations. A general covering of our service offerings. So as a company, we have multiple different areas that we can work in. We do your more standard toxicology work, we do non-GLP and GLP, we can do bioanalysis, we can do meta-ID, GCP and GLP regulatory analysis as well. And these bits will be covered in more detail further on down the line. But of course, we do in vitro ADME, we have a very strong DMPK team internally as well. We have autoimmune, CNS, which covers the blood-brain barrier, of course. We have a strong oncology team, and obviously, as mentioned earlier in the previous slide, we have a respiratory department that's headed up by a team that was formerly Pneuma Labs. So moving on to the next slide, I'll go on to, it's going to be the in silico section. I'll pass that on to my colleague, Andy Harris.
Thanks, Josh. Okay, I'll just start with a brief introduction of who I am. So my name is Andy Harris, as I've said, and I've been head of the bioanalysis group at Pharmidex since 2010, so I've been here about 13 years now. Prior to that, I spent over 20 years working for GlaxoSmithKline, GlaxoWellcome and Glaxo, working as a LCMS specialist and supporting studies in DAPK, pharmacokinetics, metabolism. So I've got sort of quite a lot of experience in that area. And since I've joined Pharmidex, it's been great to see how the bioanalysis group has grown from really quite small to its current size. And I'll sort of tell a bit more about that later on. What I'll do first, though, is actually give an overview of Pharmidex's capabilities in the sort of ADMET PK arena. So I'll just give you a quick overview of what we can do there. Okay, the first thing we're going to talk about then is in silico modelling, which is a relatively new area for the company, and it sort of arose from involvement in a grant-funded project. But basically we now have sort of dedicated full-time headcount and computational power and powerful software that can allow us to do a range of modelling. And really what we're looking to do is to actually sort of collaborate with partners in the early stage of their drug discovery programs to sort of help them in sort of maybe defining the targets and identifying potential novel pharmacophores. So we can do a sort of whole range of that sort of modelling, sort of docking molecules into sort of protein target sites. And we also have the capability to generate predictive ADMET parameters as well from potential new molecules. So this is an area that we hope is going to expand, and obviously it's sort of a very key area with a sort of general buzz around the use of computing techniques to actually enhance the overall efficiency of the drug discovery process. I'm sure this is going to be an area that's going to be of interest for everybody, and it's certainly a key area to grow for us as well. I don't have to that. Thanks. So I'm going backwards. Okay. Thanks, Ed. Okay, the company also has a strong presence in vitro ADME, so we have the capability to perform a whole wide range of assays in this area. So there's a sort of list there that summarises the main ones. So we have the sort of permeability through membranes, so we can do NDCK or CACO2 with some transport assays as well that are currently being established. We can look at metabolic stability in a range of tissues or matrices, so hepatocytes, microsomes, S9, so we can do all of those. Binding to plasma or tissues, again, it's commonly used assay using the rapid equilibrium dialysis device. We can also do profiling of CYP inhibition and phenotyping, and also determination of physicochemical properties of compounds, so things like solubility in buffers or simulated intestinal fluids, that sort of thing. We wouldn't really sort of class ourselves as being sort of high throughputs in this area in the way that some of the big companies are, but what we do offer in this area is flexibility. So we have our own protocols in place, but we're very, very happy to discuss with clients how they can be modified to suit their particular circumstances in their project area. So flexibility is one of the key things that we offer in this area. Next slide, please. Okay, move on to the pharmacokinetics area now, and this is a very big area for us. I guess it's where probably the majority of our work takes place at the moment, although that's probably changing with time. We have expertise in a number of areas here, that, as you've outlined in this slide here. So I'll start at the top right of the formulation. Basically, since it's been established, the company has carried out many thousands of studies in pharmacokinetics. And in that time, we've actually acquired a lot of expertise and experience in formulating compounds. Some clients will actually provide us with formulations and that they know work, but in many cases, it's something that we have to assess and maybe investigate as part of the study itself. And so we've actually got a lot of expertise, accumulated expertise in this area now, so we can actually help out in developing formulations for difficult to insoluble compounds, for example. We can carry out in-life studies on preclinical species on site, so we have capability to do that here. And this is live in rodent species. We can actually also do non-rodent species, but that's not that would be not on site here. These studies can involve cannulations, also a number of cannulation models available. So we can do jugular vein or bile duct cannulation, multiple cannulations as well. And serial sampling is something that we commonly do as well. So we the whole range from sort of terminal to serial, and also the sort of in between, where sort of a compromised model where it's part serial and part terminal. And we try and sort of do that to as often as possible to minimise the number of animals used in these studies. That's something we can. That's sort of study design, something we can happy to discuss with clients about how best to make best use of the animals that are available. We are also able to look at tissue distribution as well. So obviously a key aspect in the development of a compound is where it goes in the body. So we can harvest basically the whole range of organs, and also analyze them as well to determine concentrations of the drug in those organs, and really sort of determine where the compound goes in the body, particularly is it reaching the site of action. And this is what the key parameters say in CNS therapeutics, where obviously a key part of that is usually actually getting the compound to the brain and across the blood-brain barrier. So, for example, brain tissue analysis is something that we very commonly do, but we also do every other tissue as well. I think in my time here we've done just about everything you can think of. Aligned with that is how the compound gets out as well. So excretion pathways is another thing that we can follow. So we are able to collect excreta, urine, bile, and feces if necessary, and again analyse those to determine what proportion of the dose is excreted as intact parent by those pathways. We can also look at determining the structures of any metabolites that are also present in bile, urine, or less commonly feces, normally it'd be bile or urine that we look at. So this all can generate a lot of data, and one of the things that we also offer is calculation of PK parameters. So we have win-non-nin, which is commonly used pharmacokinetics software. And also an in-house package based in Excel, which is simpler and quicker to use for non-compartmental analysis, for the simple analyses. And we tend to use that for the earlier stage discovery pharmacokinetics calculations, simply because it's easier. It has been validated against win-non-nin, so we know that it gives accurate data. This means that we can basically generate the full PK profile of the parameters, and also actually help people in interpreting what that means. So we do offer the sort of consultancy in this area as well. What we find with clients is that some clients will have in-house PK experts, or they'll have external consultants that they use who will be able to interpret the data, but that's not always the case. So we can actually help with clients in interpreting this data, and also in understanding where to go with the next study, if you like. So what would be the next step in investigating a compound or a series of compounds based on the data that you've already got. So that's something that we have a lot of experience in within the company, is in this sort of project support. Next slide, please. Toxicology is an important area for the company as well. Like, this is a rapidly growing area, and as Ash said, we are accredited with the MHRA, so we can actually do these studies to GLP as well, or we can do them in slightly earlier phase as a research study. In toxicology, we can do a whole range of routes of administration, again, number of cannulations, and we can also investigate, if necessary, the relationship between the observed toxicity and the pharmacokinetics. So we'd also measure, determine the pharmacokinetics, correlate that with any observed toxicity. Organs can will be sampled, and then we can submit them for a range of testings in necropsy, clinical chemistry, histology as well, to determine any sort of detailed toxic effects. Next slide, please. And that's my area in bioanalysis. Again, the first thing to say really is that we are accredited to work to regulatory standards, so we can work for GLP and GCP as well. But we do offer the services right from the early stages of drug discovery as well in the research stage. So we basically adopt a fit for purpose approach here. The amount of checking and validation that will go into an assay is really dependent upon the stage of the project itself and how the data is going to be used. So we would generally try and discuss that with the client ahead of time and establish what is the appropriate level of qualification for the study. The majority of work is quantitative analysis. So again, we end up analysing probably mostly plasma or blood from studies, but also a whole range of tissues as well. And we have capability to analyse all those different tissues with a wide range of equipment. So we've got triple quads, most of them sort of pretty new and up to date. So the most recent ones are they've been purchased in the last 2 years. We also have a high resolution accurate mass system, which is on the bottom right there, QTOF, adjuvant QTOF, which can give good quantitative data, but it's also actually used for structural work as well. So we can carry out metabolite identification studies on compounds as well, looking at biourine or circulating metabolites. Already looking at samples from in vitro studies as well to sort of give a clear steer on where metabolic weak spots are within a series. Within the group, we've got a very wide range of experience. So we've got people who've been working in big pharma and CROs for many years, as long as me, basically. So people with similar experience length of experience to me, but with expertise in the regulatory side, with expertise in project support in sort of drug discovery, but also in method development as well. So I'm very proud of the group and its throughput and the quality of the assays that we perform. Okay, that's sort of completed my summary of the sort of MetTox area, and I will be attending the conference tomorrow, so I'll be outside. So if anybody has any questions, then so you can catch me up. I'm happy to answer those tomorrow if you can. Otherwise, yeah, get in touch electronically or by any means if you feel like. Okay, I'll now pass over to Steve, who's going to cover some of the areas of expertise in therapeutic areas. Oh, forgot about that one. Just to add, sorry, I missed this slide. This is as well as LCMS, we do have other analytical techniques that we do use. So we have expertise in the use of ELISAs, so we can actually carry out ELISA assays. And also we have a limit system that is used by a number of groups in therapeutic area support, and a small animal imaging system as well. Okay, over to you, Steve.
Thank you. Hi, so I'm Steve Mitchell. I guess my background, I was at Eli Lilly in the neuroscience research division for 23 years or so, working in psychiatric and latterly in terms of Lilly experience on neurodegenerative disorders. I joined Pharmidex in October 2020 as head of the integrated neuroscience, integrated drug discovery. But actually, I'm also running the CNS group at the moment. We have a license that allows us to investigate psychiatric and neurodegenerative disorders, covering schizophrenia, anxiety, depression, bipolar disorders. And in terms of neurodegenerative disorders, primarily Alzheimer's and Parkinson's disease, although we do have other licenses that allow us to undertake studies on multiple sclerosis and stroke, which I'll also touch on in a short while. In terms of our CNS exploits, the animal models tend to involve discrete lesioning or drug administration, modulation centrally, directly or indirectly, pharmacological manipulations, modulating systems again indirectly through, for example, administration of PCP or fructose. I mean, I'll mention a few of those later. The license also allows us to use transgenic animals. This is obviously quite an important area for looking at specific target-related effects, and also in terms of neurodegeneration, looking at animal models that recapitulate some of the features of those diseases. In terms of output, we have a wealth of experience in neurochemistry, obviously with in vivo microdialysis, which I've sort of used for many, many years at Lilly, and we have running at Pharmidex as well. In situ perfusion, again, to look at brain penetration, and a lot of ex vivo microdissection analysis techniques as well. In terms of analyzing, a lot of it is now we're moving to LC-based measurements, so it gives you a bit more, I suppose, broad spectrum analysis possibilities. There's one particular method we use, which I'll explore shortly, is in neurotransmitter profiling that allows danzylation of the sample, that allows you to measure in the same run a host of neurotransmitters. And then in terms of behavior, which we are sort of expanding, the normal sort of motor behavior, LMA, rotor rod, exploratory plus maze, but then we're also moving into spatial recognition and novel object recognition tasks to look at cognitive function. Next slide, Ash. In terms of in vivo microdialysis, I mean, I guess a lot of people are aware of this technique now. It is quite useful for looking and sampling the extracellular compartment in the brain area or tissue you've actually implanted the probe. It works on the basis that analytes will pass down their concentration gradient across the probe from the extracellular environment. It's then the flow of ACS through the probe that washes that through into collecting vials, which can then be sampled. It's always been questionable in terms of the sampling rate. I mean, most sampling rates, you collect samples every 10, 15 or 20 minutes. But the amount of information you actually get from that sample sort of increases the utility of the approach. But it certainly does give you some indication of actually what is actually going around the microenvironment of the microdialysis probe, and in particular in terms of the neuronal status and the effect of any drug given systemically on neuronal function. Next slide. So this is an example of a technique of analysis that we established while we were at Lilly, but we have running at Pharmidex as well. The mass spec expert that helped establish this at Lilly is working with Pharmidex in Andy's group as well. So this allows you to measure a whole host of neurotransmitters in a single sample. Danzylation works in the sense that it's a reaction that danzol adding to any primary or secondary amine or phenolic groups, and it tends to increase the molecular weight of the analyte. That makes ease of increases the efficiency of the separation by LC and also by detection by the mass spec. So this allows, I mean, I guess hitherto to develop to these sort of techniques, you're always having to run samples through different analytical procedures, be it fluorescence, electrochemical detection, which usually require use of more animals. But this actually allows you to run assays for all the monoamines and amino acids and the metabolites of those monoamines in the single run. And because it's mass spec, you could also look at drug levels. I mean, the drug may not be danzolated, but you're still able to pick up a signal from the drug at the same time. So it allows you to have a very nice sort of relationship, or a PK/PD relationship. Being able to measure drug in the same sample also has a value in terms of, so I suppose, validating the delivery of the drug in the consistency of dosing as well. So this is actually an example of actually ketamine, which is quite a topical drug at the moment in terms of depression and various other psychedelics, MDMA and psilocybin. This is the effect of ketamine on neurotransmitters, showing quite a profound effect on noradrenaline, dopamine, 5-HT, and histamine. Acetylcholine can also be measured, although it's not danzolated. It was able to be picked up in the same chromatographic run. And then, of course, below you can see all the effects on various metabolites as well. Next slide, please. So we're also sort of exploring more of behavioral endpoints. In particular, one particular model we're developing in conjunction with collaboration with B-Neuro in Manchester is the subchronic phencyclidine treatment to induce a sort of cognitive deficit model. I suppose it's a cognitive deficit model within as the schizophrenia context, but essentially the neurocircuitry is relevant for other cognitive deficits as well. Effectively, it's just a subchronic dose regime of PCP, where animals are administered bi-daily for 7 days. Then they're left to wash out for 7 days, and it produces this prolonged deficit that can last for weeks and several months as well. The group in Manchester used novel object recognition as a way of, I suppose, assessing the cognitive effects of this treatment and any sort of pharmacological treatment to restore cognition. And I guess this utilizes the natural sort of behavior of an animal to explore novel environment or novel objects. So they're actually in the acquisition trial, they have 2 identical objects. Then they're taken out and put back into the arena where one of those objects has been replaced. And the idea is that that animal would normally explore that novel object more than it would have done the familiar. And this is what's clearly seen in the blue column, the blue graph. And that sort of ability to distinguish novelty is lost in the animals that have been treated with PCP. Next slide. On the same sort of vein, I guess, spatial recognition task, again, it's a feature of the animal to explore novelty. And again, it's just the animals are exposed to one compartment, and then on the day of test, 2 compartments, they allow access to compartments, and it's how much time they spend exploring the novel compartment, the one they haven't been used to visiting. And certainly in vehicle-treated animals, they show a clear distinction between novel and familiar compartments, where they explore the novel compartment more than the familiar, which is lost in this particular exercise with scopolamine, and the condition is restored with donepezil treatment as well. So these are sort of 2, I suppose, models that allow you to assess cognitive function. The first model is quite useful in terms of having a prolonged cognitive deficit, with which you can then look to see the effect of any sort of modulatory pharmacological interventions on that deficit. The SRT, we are exploring the effect of PCP in this model as well, but it can also be used for acute pharmacological deficit, as in this example with the reverting scopolamine-induced deficits. Next slide. So there are also, as I mentioned at the beginning, there are two other areas that we are able to investigate with different project licenses. One's on multiple sclerosis and on stroke. So next slide, please. So EAE, experimental allergic encephalomyelitis, I mean, I suppose it's the most common model for investigating MS. As you know, MS is a sort of chronic demyelinating inflammatory disease. It's supposed to be an autoimmune condition. And what in this particular model, it's quite useful for investigating those neuroinflammatory elements that may be associated with the disease. The pathophysiology of this model is based on the reaction of the immune system to brain-specific antigens that are administered. The reaction induces like an inflammatory response that leads to loss of myelin and neuronal degeneration, and the consequent sort of neurological symptoms that are commonly seen in MS as well. In terms of the brain-specific antigens, we use MOG, which is myelin oligodendrocyte glycoprotein. So these animals are immunized with a subcutaneous administration of MOG and CFA, which is helps to boost the immune response. They're also administered pertussis toxin, that again administered to induce a sort of more of an immunological response to the MOG administration. And then after about 9 to 14 days, you get the development of these clinical symptoms that sort of start with sort of a mild loss of tone and to eventual paralysis, starting from the tail, the hind paws, and then the forepaws. And this is a model that can be used to look for drugs, as I said, that more, I suppose, more related to the sort of neuroinflammatory component rather than the ongoing degeneration. Next slide. As I said, we have ability to look at stroke. This is around the sort of lesions or blocking the middle cerebral artery in the rat or mouse. There are two approaches. You block the MCAO either transiently with a filament or permanently with the coagulation model. We tend to use a transient ischemic model, the filament model, where you have a 45 minutes occlusion, and then you get reperfusion, and you look at either the amount of the infarct size using TTC staining, or the consequent effect on various behaviors, motor degeneration, various neurological scoring using the Garcia or Bedesson scale. You can also look at allodynia and thermal hyperplasia, and of course you can look at histological staining at the end using TCC, and look at biomarkers as well. Next slide. So unfortunately, Chris Ireson isn't available to sort of present this area. He has a wealth of experience in oncology in academia and industry, and he's actually written an article on the role of mouse tumors models in drug discovery for anti-cancer drugs, which I sort of recommend as a useful reading material. We have a range of animal models, mostly around subcutaneous administration of cell lines derived from either human or mouse cancer cells or mouse tumors, primarily injected subcutaneously into the flank of the animal, or orthotopically into the organ from which the cancers develop. Alternatively, we can also use PDX, which is the patient-derived xenograft, which is actually freshly derived from human tissue. But I'll show you the most predominant methods are the cancer cell lines. The models are useful in the drug discovery phase for ensuring that molecules have the appropriate pharmacological activity, in the biological system. The models provide a measure of the functional modulation of the tumor in vivo, with which we can actually then call it as I guess a pharmacodynamic endpoint, which can then be used to develop a PK-PD relationships. The models also provide an opportunity to have some preliminary insight into toxicity, as well as measuring the size of the tumors as they recess with treatment. As I said, as I mentioned, Chris has a sort of vast experience in this area, and his expertise has been quite helpful in terms of with clients in terms of the data evaluation and interpretation of the findings. Next slide, please. So this is like a list of the models we have across a whole range of different indications. Essentially, as I said, using administration of cell lines from either human or mouse, given orthotopically either into the organ of origin or heterotopically into the subcutaneously into the flank of the animal, and they cover a whole host of different cancers. Next slide. Chris and his team have been very effective at gaining, I suppose, grants and external collaborations for a whole host of different cancers and also modes of delivery of anti-cancer therapies. This is a list of grants that have been achieved that are ongoing and also more recently as well. The important thing is that they were with a whole host of different number of partners. So that sort of shows the breadth of experience and expertise within this area. And they also provide a certain amount of financial gain to the company as well as increasing our sort of profile and networking externally. Next slide. So that's I'd like to hand over to Ian now, talk about respiratory diseases.
Okay, right, thanks, Steve. So yeah, just as a way of introduction, so I'm Ian Knowles. I guess as a background, I've been working in contract research for just over 20 years, having joined a company called Neurolabs, who were founded in 1997 as a respiratory-focused contract research organisation. And as Ash mentioned at the outset of the presentation, Pharmidex acquired Neurolabs back in 2018, and in essence, most of the sort of therapeutic capabilities in the respiratory space were moved across to Pharmidex. So the presentation, in terms of obviously respiratory perspective, really going to hopefully give a snapshot of some of the main disease areas we're focused in. So these are bullet pointed in this first slide here. So most of the presentation really focuses on the top 5 sort of bullet points, but just to obviously add in that we do have models in terms of obviously in vivo capability for looking at allergic rhinitis, which is induced by giving allergens to mouse or guinea pigs. We also have been heavily involved in the respiratory infection space in the last 5 or 6 years. Obviously, it's become incredibly trendy, obviously with COVID, sort of pandemics over the last 3 years or so. So we have capabilities within that area, which obviously we can discuss separately if there's an interest. And we also have a standardised model where everybody, generally speaking, has bleomycin models as a means of inducing pulmonary fibrosis. So we're no different in that respect. Our main, I guess, novelty factor is that we also have guinea pig models looking at fibrosis as well. This allows the additional endpoints where, in pulmonary fibrosis, a lot of patients develop chronic cough at a later stage, and this allows us then to actually model this within our laboratories. So actually, if you move on to the next slide. Okay, so moving then on to the main sort of asthma area. So I guess this certainly has been one of our main drivers for the last 20 years in terms of the model development area. And I guess we've divided our models really into sort of 2 main areas, where you're looking at models which mimic a lot of mild asthma. And then generally speaking are acute models, so operate over a sort of 20 to 30 day window. And then we also have models of more severe or asthma or exacerbations of asthma. So this slide obviously highlights the more acute mild asthma. And we've set up a variety of models in different species, so most sort of rodents, so obviously the typical ones being mouse, rat or guinea pigs. We also have models set up where we've looked at allergen challenges in rabbits as well. And I guess depending on obviously drug targets and what phenotypes of asthma you're looking to model will then dictate what allergens you may want to look at. So for example, with house dust mite, we can challenge different rodents with sensitization protocols, which then model certain either Th2 and Th17 inflammatory pathways, which then models your eosinophilic and neutrophilic asthma phenotypes. Ovalbumin is more of a specific one for looking at Th2. So if your drug targets are purely for taking out Th2 inflammatory pathways, then that would be a model that's more relevant for looking at efficacy. And more recently, in the last 2 or 3 years, we've looked at Alternaria, which actually activates Th17 pathways along with Th2 as well. So in most of our studies over the years, we've then obviously profiled a lot of positive controls. So obviously in the asthma space, being able to demonstrate the effects of steroid treatments is quite key. So we have background data looking at the effects of dexamethasone, budesonide, and fluticasone. Again, which one of those you may look at in your study might be dictated by route of administration and where you see your potential drug mechanisms being delivered. So, for example, fluticasone can be delivered directly into the airways. So, if you're looking for aerosol treatments, fluticasone might well be the ones to compare against. Obviously, this goes through phases where PDE4 inhibitors have been quite trendy. So, we have background data where we've looked at Flumalast. Again, also with leukotriene antagonists, so montelukast, we've got validation data for. And then, obviously, because of the inflammatory process and some of the newer medications that are present in asthma, looking at sort of antibodies for taking out Th2 interleukins is obviously relevant. So we have looked at anti-IL-5, anti-IL-4, and anti-IL-13. So all of these are sort of relevant to look at effects as potential treatments. So most of our models follow a similar endpoint analysis process. So you'll see commonality amongst the sort of models of what we would obviously can offer. So within asthma, what you'd expect to see is the airways become hyper-responsive to neurotransmitters. So we would offer as a standard endpoint looking at functional change within the lung. And you can then look at this by challenging animals with methacholine, and you get an increased constriction from a normal airway. And this would also be very relevantly typical to what asthmatics would experience. Generally speaking, we tend to use most Bucksco systems for this, so some of the latest technologies within the sort of area at the moment. Alongside this, obviously, mostly asthma being an inflammatory condition, one of its key primary endpoints obviously then looking at inflammatory cell populations. And this can be done by looking and taking fluid from the lungs, or we can take lung tissue and look at increases in sort of cell population of the tissue itself, and also to look at sort of cell levels within the blood. And this can be done in terms of obviously looking at total cell numbers and differential cells. So if you then look at the eosinophilia or neutrophilia to demonstrate that the models are then demonstrating these sort of phenotypes of asthma that you'd be expecting. Alongside this, as we mentioned earlier, we have capabilities of doing ELISA and Luminex, so it's relatively straightforward for us to then look at specific biomarkers to demonstrate that the Th2 or Th17 pathway is being activated. Again, we can also do histopathology, so this can be utilised for looking at sort of cell numbers migrating into the lungs as one of the sort of primary endpoints. And as evident from the early part of the study or sort of presentations, we also have capabilities in-house of doing the bioanalysis. So alongside all of what we can do, we can take serial bleeds or terminal bleeds to then do potential bioanalysis in-house as an add-on to these studies. So Ash, if you'd like to move on to the next slide. So this really just gives you a snapshot, in essence, of data from a particular studies that we've run. Primarily really demonstrate the difference between mild asthma, which is on the left-hand side, sort of data sets, and a more exacerbated severe asthma model, which is illustrated on the right hand side. So both of these are sort of different models looking at house dust mite affecting the mice, and this is lavage fluid taken from the animals following sensitization and challenge. So if you primarily just focus on the left hand graphs first of all, on the top you have total cell numbers within the lavage fluid, and then at the bottom you have eosinophils. So these basically eosinophils being driven by Th2 pathways. So in the green columns on the left-hand side of each graph, this shows you the standard effect we'd expect to see. So you've got naive animals around the effect of house dust mites, vehicle-treated animals, so you're getting a significant increase in total cell numbers migrating into the lung tissue or lung space, which is picked up in the fluid. And then with treatments with dexamethasone, you can get a significant inhibition of these total cell numbers, which is obviously what you expect to see within a clinical space, but steroid treatment would inhibit this inflammation. And this is primarily driven by the eosinophils, as shown in the left-hand, bottom graphs. On the right-hand side, this then shows you the subtle difference between severe asthma and the fact that, obviously, from a clinical perspective, steroids in these exacerbations and with more severe asthma, the steroid, in essence, starts to have less sort of clinical effect. And this is illustrated in our influenza exacerbated house dust mite model, where you're looking again, if you sort of look at first left-hand columns in green, it shows you effects of house dust mite with deactivated virus in the light green, and then with an activated virus, influenza virus, in a slightly darker green. So you get a significant increase with the virus treatment. And with dexamethasone treatments, even going up to concentrations higher than what you'd expect to see working in a mild asthma model. In essence, the dexamethasone treatments fail to have any sort of clinical or significant efficacy. And this is obviously illustrated then as well with the eosinophils. So the Th2 pathways are fully activated, and dexamethasone steroid treatments fail to inhibit it. So if we move on to the next slide. So that really just gives you a bit of a snapshot overview of asthma. Obviously, we've got a lot of background data that can be shared. And obviously, if there's interest in particular models and different sort of allergen challenges. Another area we've been heavily involved with is using models to induce acute lung injury. So these are quite short studies, ranging from anywhere from about 4 hours of treatment times over to 24 hours. So the turnaround time of the study is very quick. And this induces a model of generating neutrophilic inflammation in the lung. And this is generally induced by endotoxin exposure as a standard, which in our hands with the profile for E. coli and Salmonella. And this can be administered to, again, to the usual rodent species to look at, so mouse, rat, and guinea pig. Alternatively, mediators we sometimes also get involved with using is ozone. So the advantage of ozone is it mimics a little bit more of a COPD type model. So it translates more effectively to as an early screen to COPD studies, depending on what your program of research is. We can also use poly-IC, which acts as a pseudo-infection type model, but again causes acute lung injury as mimicked by inducing this neutrophilic inflammation in the lung. So in these models, you'd expect steroids to work, and again, we've obviously profiled the usual layer of steroids, but in the clinical space. So there's background data demonstrating their efficacy in these sort of lead mediators. And as depending on how trends went, obviously there's background information on the PDE4 inhibitors, which certainly have a sort of resurgence of interest in the last 5 years or so. Again, the same endpoints sort of profiling, so inflammatory cell populations in the lavage fluid can be evaluated. We can also look at the lung tissue, again using sort of histopathology to look at sort of cell migration into the lung, and also looking at blood to see the increase in sort of cell populations. Obviously, biomarker assays using Luminex is obviously an additional endpoint which sort of bolt onto these particular studies. And again, like with all of the models, we can then look at the bioanalysis, so we can take kind of serial or terminal bleeds and then pass on the samples to our bioanalysis team to look at. Move on to the next slide, please. So this then moves on, I guess, to the COPD models we have. So I guess certainly in my time being involved in respiratory contract research, a lot of organisations will use the acute lung injury endotoxin models as an early screening model, and then move into more a disease modelling for COPD. And traditionally, over the last 20 years, most people have looked at using cigarette smoke to induce a model of COPD. So the idea being that the cigarette smoke induces this neutrophilic response, which is steroid insensitive, and it's critical to be able to demonstrate this sort of lack of response to the steroid in the model. Traditionally, we tend to use dexamethasone as our steroid, where you basically, in essence, don't see any efficacy with this particular sort of steroid treatment. So we have access to 3 models. Basically, it was a 4-day acute cigarette smoke model. So this allows time for neutrophil influx into the lung, which can detect them in the large fluid, as well as in the lung tissue itself. 10-day model then starts to show slight more changes in sort of mucus levels, neutrophilia, etc. And then we also have a 3-month chronic model of cigarette exposure, and this then tends to then lend itself nicely to looking at the emphysema aspect of COPD. And obviously, I think in terms of obviously driving this sort of program of research, then it makes sense to do this in a stepwise fashion, unless your drug treatments are purely looking to prevent emphysema itself. More recently, we've also looked at using ozone as an alternative approach from cigarette smoke. The advantage of ozone is that you get a nice, robust sort of neutrophilia in the lung, which is steroid insensitive, but then also removes the complicating factors of any sort of pollutants that are obviously in cigarette smoke, which then can interfere with data sets potentially and interpretation of data. So I think there's a lot of a trend to move away and start to utilise ozone as an alternative modelling for COPD. So if you can move on to the next slide, please. Okay, and so another area just to touch on, obviously a background, I guess there's also been an interesting cough, certainly from our perspective as a company, to complement a lot of the respiratory diseases. So all of these diseases, COPD, IPF and asthma, have a chronic cough as an underlying component as the disease progresses. So obviously there's an interest in developing treatments primarily focused on the chronic cough of these diseases. So we've had an interest in this space. So we have a number of models set up at the moment. Most of the research is carried out in guinea pig models. So they will spontaneously cough to tussive agents. But we've also, certainly I have in the past, looked at using rabbits, where you can expose them to ozone, which you'll see down there as an exacerbating agent. And then you can ensure that rabbits don't cough to things like citric acid. And then the subtle variations in the neural setup of rabbits to humans, which makes them relevant depending on your drug targets. So there is definitely a space for rabbits in sort of cough research. Standard mediators, as well documented that citric acid is used to induce tussive responses. Depending on whether you're looking more purely at drugs that may interfere with C5 activation, then there's a potential role for capsaicin as a mediator. To then model more of an exacerbation of chronic cough, we've then more recently set up models where we're looking at the actual disease component. So you're setting up, for example, an asthma model and then looking at tussive activity. So in our hands, we've looked at ovalbumin's Th2 mediated asthma. You get this eosinophilic asthma, but then looking at the tussive agents. And the same can be done with bleomycin. So you can basically expose guinea pigs to bleomycin, reduce the fibrosis, and then you can either trigger tussive activity with citric acid or capsaicin, or look at spontaneous activity. Other agents can be used, like I mentioned, ozone. Obviously, I've used up, so we've used up for triggering tussive activity in rabbits, and equally as well, histamine can reset the sort of sensitization in the vagal nerve in the lungs to induce cough. And this has become relevant with the massive interest in P2X3 antagonists drug treatment for cough. So we've validated models in that area. Positive controls in this space, historically we've always used codeine, acts centrally and peripherally to take out the tussic response. The same can be done to a certain extent with lidocaine, taking out peripheral mechanisms. More recently, in the last 5 to 10 years, I guess, obviously P2X3 antagonists have become quite a trendy area of development. This is first triggered by Pharma, who had a formulation AF219, which I believe was then acquired by Merck, and hence one of their treatments for activity. So we have background control data demonstrating effects of these P2X3 antagonists. Standard endpoints obviously looking at total coughs in a certain exposure period to a citric acid or capsaicin. You can then look at changes in frequency of cough, see whether there's any changes in pattern. So normally you see, generally speaking, you see an increased activity in the first period of time, and then it sort of stackers down a little bit, so you can see if that profile changes. And latency to offsets, so if the neuronal drive is being inhibited in a way, you get changes in cough latency. And as with anything, we can also then take terminal bleeds and use those for bioanalysis, so levels. So on the next slide, which I think is the last one that you spoke to, it showed you some background data model. So this is just citric acid effects. So in our standalone citric acid model, if they're exposing animals for 10 minutes to a 1 molar concentration as an aerosol, and the green bars on the left-hand side, you can see you'd normally expect in this 10-minute period something in the region of about 30 coughs. Now, if you treat the animals with codeine 2 hours beforehand, so 25 mg per kg IP, then you get a significant inhibition of this cough response, which is a good starting model to utilise. Obviously, the one thing that then, in terms of obviously modelling the cough becomes relevant, is obviously this has a protective nature. So you don't necessarily want to be looking at models which totally take out the cough response. Hence, looking at models which are more clinically sort of relevant, hence the exacerbation of cough models. And this is illustrated on the right-hand side. And this is showing the effects of histamine exposure. So it's mimicking a little bit what you'd expect to see, say, in asthmatics, where there's mast cell granulation and histamine, ATP, etc., mediators of that nature, and resetting vagal drive. So in this model, the green bar shows you a typical response in an animal that's just exposed to citric acid. You normally expect to see about 6 coughs in this particular model at a slightly lower concentration of citric acid. With the histamine pre-treatment for 2 minutes, you're going to get this massive increase in cough response. And then with drug treatments for codeine, again, you can inhibit through central inhibition mechanisms the actual cough response. And this is also true with the P2X3 antagonists acting peripherally. You can inhibit the exacerbated tussic response. So that's a bit of an overview of the respiratory area. So slide.
Yes, just to round off the presentation in general, Pharmidex as a company operate in 3 specific areas, but we offer fee-for-service, like most CROs. We also do joint collaboration, which is going to be the grant funding route. So we've been particularly successful in this area recently. At the moment we have 3 Innovate UK grants actively. In this round and last call we had 2 as well, so we've been very successful in this area. So we're happy to work with companies that have novel ideas, need our support in terms of actually conducting the work to work together on a joint application. And we have shared risk, shared reward, which is another model that we operate in, which is if you come to us with an idea or we have an idea ourselves, we'll approach people and we will share the risk and share the reward in terms of IP and so on and so forth going forward. So we are quite unique in that respect in terms of we are able to work with people in multiple different avenues, and that's something that we're quite keen to do. So if that is of interest to you or your company, please do contact us at bd@pharmidex.com and we'll get back to you to discuss some further terms. And that rounds off the presentation for today. Thank you very much.
Excellent. Thank you so much to the Pharmidex team there. We are up to time, but before I sign us off, does anyone from your team want to say one word on why you should be the partner of choice for anyone looking for your expertise?
Of course, yeah. I think as a
I think we probably all have something there. I think for me, we've got a highly experienced team, we've got a great depth of experience within the company, and there's a great commitment to delivering quality science to our clients. You know, I think we're not just here to provide a service, you know, sort of hand over data. We actually want to engage with clients, understand their projects, and also work with them as partners rather than just a simple a simple service. So I think that's, you know, that close relationship really actually helps in the progression of compounds through projects. And I think that's been demonstrated with a number of our clients who had been with us for a long time, and we've actually seen, you know, the compounds go through to the clinic. So we've actually got quite a bit of success in that area.
Absolutely. I think, you know, the nature of the businesses we've developed over time is quite bespoke. We're happy to work on different projects, different endpoints. We're very flexible as a company, and we'll never overpromise and underdeliver, which is the main thing as well. We will do exactly what we say we're going to do, and we'll also work with our clients for the best outcome. There's no set structure here. We very much want to work within what your endpoints are and how best we can achieve them as a company.
Excellent. Thank you. Fantastic thorough presentation. Thank you so much to the team. Thank you.